audible? Yep. Om Shri Sai Ram, welcome back to the next session of International Microorganisms Day, and most importantly, the most prestigious and the most important Dr. K. Gopinatha Rao Memorial Lecture. Before I go ahead and introduce the speaker, I think it is befitting the occasion that I give a short introduction about Dr. K. Gopinatha Rao. Dr. K. Gopinatha Rao was born on September 12, 1928 into a family of physicians. His father, Dr. K. Vasudeva Rao, was a well-known physician in Madras and the second superintendent of the famous Tambaram Tuberculosis Sanatorium, while his maternal grandfather, Dr. V. Srinivasa Rao, Resident Medical Officer, Government General Hospital, Madras, had the rare privilege of helping to care for Sri Ramana Maharshi in his later life. Dr. Gopinatha Rao's parents prized education and service to others, and from a young age, he observed his mother, Srimati Saroja Bai, and his father, Dr. Vasudeva Rao, model inclusion and generosity in their large joint family. Dr. Gopinatha Rao imbibed these qualities in great measure. Following his doctoral training from various institutes, Dr. Gopinatha Rao settled in Canada with his wife, Srimati Kala, and 3 children, where he practiced as a urologist, first in Brandt, then in Manitoba, and then in Cornwall, Ontario, Canada. For more than 3 decades, he cared for countless patients with great skill and dedication. In addition, he was passionate about advancing the field of urology and published several articles in notable medical journals and journals of international repute. In 1992, he was blessed by Bhagwan Sri Sathya Sai Baba, founder chancellor, to relocate to Puttaparthi and join the then nascent urology department of Sri Sathya Sai Institute of Higher Medical Sciences in Prashanti Gram. For his children and family, Dr. K. Gopinatha Rao was a beacon of compassion, tolerance, humor, and for his colleagues and patients, a meticulous surgeon of great talent and deep humanity. In memory of this great person, today we are going to have a memorial lecture that is going to be delivered by Professor Roland Brandt. Professor Roland Brandt has established himself as a leading figure in the field of neurobiology, with research that has made profound contributions to our understanding and development of aging, neurodegenerative diseases, and degeneration of nerve cells. His work spans molecular, cellular, and systematic levels, providing a holistic perspective on neuronal function and dysfunction. A major focus of his research is around Alzheimer's disease and even the central part of this Alzheimer's disease, that is the tau protein, whose misregulation plays a critical role in progress of neurodegeneration, which was being absorbed by Professor Brandt's group by employing state-of-the-art live cell imaging techniques, which will allow them to visualize in real time the dynamic processes that underlie neuronal decline. His research program is both broad and highly specialized. Key areas of investigation include the molecular mechanisms of Alzheimer's disease, the role of cytoskeleton in the development and aging of neurons, and the cellular pathways that mediate stress responses. To pursue these questions, his team employs diverse model systems such as neural cell cultures, organotropic brain slices, and transgenic mouse models, ensuring that discoveries are mechanistically rigorous and biologically relevant. Professor Brandt's methodological expertise is equally remarkable. His group integrates a wide array of advanced approaches, including cell and tissue culture techniques, viral and non-viral gene transfer, algorithm-driven image analysis of live cell imaging data, which is state-of-the-art facility, and extracellular recordings using multi-electrode arrays. This technological sophistication allows his laboratory to generate insights that bridge basic biology with disease mechanisms, often illuminating new possibilities for disease intervention and developing new therapeutic strategies. The impact of his work is evident in many influential publications. His studies have revealed, for instance, how chronic presence of oligomeric amyloid beta alters synaptic structure and function in Alzheimer's models, and how RNA binding proteins partition dynamically into something called as stress granules under conditions of cellular stress. These findings not only deepen our understanding about the mechanism and the advancement of neurodegenerative disorders such as Alzheimer's disease, they also provide crucial footholds for translation research aimed at combating such neurodegenerative diseases. In sum, Professor Roland Brandt's research stands at the forefront of modern neuroscience, exemplifying scientific rigor, innovation, and vision. Through his pioneering work, he continues to inspire both the scientific community and next generation of researchers committed to advancing our knowledge of brain, health, and disease. Without much further ado, I request all of you to join me in welcoming Professor Roland Brandt to deliver Dr. K. Gopinatha Rao Memorial Lecture.
Very kind introduction. Also, thank you very much for inviting me here to this beautiful place. I really enjoyed the hospitality also from Professor Vendekesh, and I'm impressed what I was seeing until now with respect to the possibilities which you have here in doing state-of-the-art research. So I'm coming from the University of Osnabrück, which is in the northern part of Germany. And you can see on the slide here the science campus of the university. And actually the biology building is here in the back of the campus. And much of my work, or the work of my group actually, has been done in the Cell Nanos Institute, where high-end microscopy is provided, which allows, for example, also single molecule tracking of molecules, and this will be part of my presentation. I not only want to present the techniques behind, but mainly for what they are useful with respect to analyze questions which are related to neurodegenerative diseases. So what I want to do today is actually to first give you a short introduction into the tau protein. Tau stands for tubule-associated unit. Its role in health and neurodegenerative diseases, so-called tauopathies, and one of the most common tauopathy is Alzheimer's disease. Since this is a congress on microorganisms, I would also like to include a short section on how it is possible and how it can be useful to study tau biology in yeast, in Saccharomyces cerevisiae. And we did some work together with a collaboration partner in that respect. Then I would like to move towards the mechanisms of tauopathies. As the title already promises, I would like to spend some time on explaining the kiss and hop mechanisms of tau's interaction with microtubules and how that can be related to neurodegenerative diseases. And at the end, I would like to move forward towards the development of new drugs against tauopathies and how these imaging approaches can be used to identify compounds which are addressing a key component of the disease, and in this case, it is a tau protein. So if you feel that you want to interrupt me and ask a question, don't hesitate. I'm giving also lots of lectures for students, and I always tell them, try to get me out of my presentation, and I will do my best then to answer your question. So there are a couple of diseases, many diseases actually, which are characterized by changes in the tau protein, and they are collectively called tauopathies. And the changes which are observed are an increased aggregation of the protein, which you can see here on the right side. This is an electron microscopic image of tau aggregates isolated from a patient with Alzheimer's disease. And these are actually paired helical filaments. You can see here that these filaments are actually showing a regular turn, and they are composed of tau protein. And another feature, which we will also discuss a little bit later, is an increased phosphorylation of the protein. So there are a whole list of tauopathies currently known. I don't want to go through all in detail because that would simply be too boring. But one disease is actually quite informative for tau researchers, which is frontotemporal dementia with Parkinsonism linked to chromosome 17 or FTDB17. Why is it important? It is important because it is a primary tauopathy. And primary tauopathy means that changes in tau protein alone are sufficient to cause the disease. In this case, these are mutations. So a mutation of tau can be axonic, can be intronic, leads to tau aggregation, and leads then to the development of the disease. And actually that was the first proof that changes in tau protein are sufficient to cause the disease. However, the most common tauopathy is actually a secondary tauopathy, which is Alzheimer's disease. And secondary tauopathy means that tau changes are occurring, but they are not considered to be the starting point. The starting point appears to be another protein which is aggregating in this context, in the context of Alzheimer's disease, that is A-beta. So it is assumed that A-beta is aggregating. This aggregation of A-beta is then causing changes in tau protein, increased phosphorylation called hyperphosphorylation, and increased aggregation. So we will come to that in a second. However, I would already like to introduce now that this is actually the so-called amyloid cascade hypothesis, which mentions that A-beta aggregation comes first and that causes and changes on the level of tau. Nevertheless, it is a tauopathy because, and it's becoming more and more obvious, that actually the changes in tau are the factors which kill the neurons, therefore they play a central role in the disease process. So, Alzheimer's disease is a very common disease. It is very common in the elderly population. So according to estimates, by the age of 80, 1 out of 3 individuals will be affected with Alzheimer's disease. So we are speaking about extreme number of Alzheimer's disease patients. And actually the first Alzheimer's disease patient was Mrs. Data, which was a patient of Alois Alzheimer, who also gave the name to this disease. And Alois Alzheimer was also able then to look at the brain of the patients after she was dying, and what he was observing, and what since then was observed many times, is that the brain shows characteristic changes. It shows 2 types of aggregates. One are the so-called senile plaques, which are composed of amyloid beta, which I was already mentioning before. These senile plaques are outside of the neurons. And the other pathological change are actually tau aggregates, which cause these neurofibrillary tangles, which are inside the neurons and which are the basis for Alzheimer's disease to be tauopathy. So, let's do it that way. So here you can see a graphical representation actually about, no, you can't see it because it went one too fast, like that. Which is showing this amyloid cascade hypothesis, that actually the starting point of Alzheimer's disease is a formation of amyloid plaques consisting of oligomeric A beta. Nowadays, it is assumed that actually the oligomeric form, the soluble form of these aggregates is a toxic species. And these oligomeric A beta then induces changes in the tau protein. It leads to the formation of oligomeric tau aggregates, which then develop further towards these neurofibrillary tangles, which you are also seeing here in this slice. Oligomeric tau is actually then the trigger, who is, or the executor of a neurodegenerative triad together with A beta, leading to the impairment of synapses. Synapses are lost. These are the communication sites between the neurons. Dendritic simplification, so the complex array of dendrites becomes more simplistic, and a loss of the neurons and a loss of the axons, and all of that is considered then to cause dementia. So what you can see already on this representation is that tau actually plays a central role by triggering this neurodegenerative triad. So currently the first treatments for Alzheimer's disease are trying to tackle this oligomeric A beta, like this antibody treatments, which are now newly on the market. And this is actually the first causative development with respect to treatment of Alzheimer's disease. However, you can see here that actually a target which is equally well suited would be tau and the tau oligomerization, and that is why we and also many others are concentrating on tau and modulating tau aggregation and hyperphosphorylation. So let me introduce tau a little bit, since I think that most of you don't know it in detail, but it helps us to understand the later points of this presentation. Tau is a microtubule associated protein, a MAP, and is specifically expressed in neurons. It is interacting with microtubules through its microtubule binding region, and as you will hear later, the interaction is highly dynamic. So it shows a complex, developmentally regulated expression pattern. So in humans, we just have one tau gene, but due to alternative splicing, many proteins are formed. In the central nervous system, these are 6 different isoforms, dependent on alternative splicing of 3 axon 2, 3, and axon 10. In the peripheral nervous system, 2 additional isoforms, which are much longer, are produced, and these are called big tau because of their larger size. So in addition to these different isoforms dependent on alternative splicing, there are also diverse post-translational modifications occurring, and the best studied modification, post-translational modification with respect to tau is phosphorylation. And these are some of the phosphorylation sites which have been identified in tau, and you can envision that also that produces a significant complexity, since many of the sites can be phosphorylated. So, but it's not only phosphorylation, there's also acetylation, O-glycosylation, ADP ribosylation, which can occur on the level of tau as post-translational modifications, but the best studied, and also with respect to correlation to the disease, perhaps the most important one is phosphorylation. So we could already show many years ago that tau is actually the first marker for the development of an axon. So when we look at the localization of tau protein in cultured neurons, in this case cultured hippocampal neurons, then you can see there, here that very early during the development of polarity, when an axon actually is developing, tau is already localized here at the distal axon. At a state where another microtubule-associated protein, MAP2, which is later only present in the somatodendritic compartment, is still present in all of the processes. So in that respect, tau is one of the earliest markers of the development of polarity. And later, during the development, if polarity is developing further, then you see that tau shows a characteristic proximal distal gradient in the axon, and the concentration of tau is actually highest here at the transition between the axon shaft and the growth cone. So, I always have to point in this direction. What happens during disease? You just heard that tau is a protein which is enriched in the axonal compartment. That is due under physiological conditions. During disease, that changes. During disease, we see a redistribution of tau from the axonal compartment into the somatodendritic compartment. This redistribution is associated with an increased phosphorylation of tau, which is called hyperphosphorylation in the literature, and by the aggregation of tau, forming paired helical filaments, as I was already introducing them before. So how these changes are related to each other, whether first tau gets increased, shows an increased phosphorylation, and then redistributes or aggregates first, all of that is actually still not completely clear, but of course it's very important also to understand the disease mechanism and also to develop drugs which interfere with these pathological processes. So on the left side you can, for example, see, I have again to go back, yes. On the left side, actually, you can see the distribution of tau in a patient with Alzheimer's disease. You can see that tau, in this case, we stained phosphorylated tau, is practically only present in the somatodendritic compartment, not longer in the axon which would protrude here from this pyramidal neuron. And this is also evident here, and what you can also see here very well is that this mislocalization and aggregation of tau is associated with the degeneration of the neurons, that the processes are not straight any longer, but they show this curved morphology, which is also a sign for the neurons that they are affected, that they are on the verge of dying and degenerating. So the important aspect is during disease, and actually early during the disease. One should also mention that tau is actually a chronic disease which takes decades to develop. Tau redistributes and aggregates in the somatodendritic compartment. So I promised you that also I will have a connection actually to the microbiology, which is due to a collaboration which we did with Professor Heinisch, who is also at the University of Osnabrück, and he leads the genetics department. And actually we were writing together a review article on the signaling pathways and post-translational modifications of tau in Alzheimer's disease, as it can be studied in a microorganism in yeast. And you can do lots of different things in microorganisms, like in yeast. It's a beautiful tool, which you cannot actually do in cells, or if you do it, it's very laborious and time-consuming. So this is a list. For example, you can do complementation or deletion studies. What we did, actually, we did a heterologous gene expression. We were interested in mechanisms, how tau is localizing to the axonal compartment, and we could identify at an earlier time that tau is interacting with membrane components, and the membrane components which apparently played a major role was annexin. Annexins are proteins which interact with the plasma membrane, and they do that in a calcium-dependent manner. And we could also show that annexins are also interacting with tau proteins, so probably producing a link between the plasma membrane and the cytoskeleton, the microtubule skeleton, via tau. So what we wanted to do in a yeast system is to understand better and also do a functional mapping of the interaction of tau with annexins. And actually, that we did together with Jürgen Heinisch and other members of the laboratory. And the question is, why did we, for the HAL, use yeast? Actually, for several reasons. One reason is that neither tau nor the annexins are endogenously expressed in yeast. So we can somehow humanize yeast by expressing these proteins and then checking for the interaction with human proteins without having to care about the endogenous yeast proteins. Second, we found out earlier that if we want to analyze calcium regulation, that is not possible in a mammalian system because in the absence of calcium, actually the annexins tend to unspecifically aggregate, which is not the case in yeast. So we can also study calcium regulation there. And the third point is that tau, for any reason, does not bind to yeast tubulin. That is also important because if tau, for example, binds to tubulin or microtubules, you have a problem to study direct interactions because the interactions could also be indirectly. Tau via microtubules to any other protein. And that can be, with such an approach, excluded actually in a yeast system. So the first experiment, of course, was to analyze whether annexin is interacting with tau. We expressed both also a control protein with a tag, this was a GFP, in yeast. And we could show, as you can see here, that in the presence of calcium, tau is interacting with annexin A2 because we can pull it down. If we do the same experiment without calcium, tau is not longer being pulled down, which tells us, okay, we need calcium in order to have this interaction. So tau and annexin A2 interact in a calcium-dependent manner. That is already what we can learn out of that here. We can then perform actually a functional mapping of the interaction. We can simply go through several deletion constructs of tau. We can then analyze what is required for the interaction with annexin. And just to cut a long story short, you can see here when we have a very short tau fragment, which actually just encompasses here the extreme end terminus of tau, which is protruding from the microtubules when tau is binding to microtubules, that this is sufficient to show an interaction with annexin A2. So the interaction appears to be mediated by axon 1 at the extreme end terminus. So then we can check what is on the level of annexins going on. And if we do similar experiments, then we can, for example, check the end terminal region of the annexins or the core domain of annexins. And when we check for the N-terminal region for amino acids 1 to 34, then you can see tau is not pulled down. However, when we take the core domain, then we can see the interaction with tau. So that tells us that tau is actually interacting with the core domain of annexin A2, and that dependent and occurs via the extreme N-terminus of tau. So this is interesting because actually annexin A2 is a member of a whole family of annexins. And the main difference between the annexins is the N-terminal region here. So since tau interacts with the core domain, that would mean that it's not only interacting with annexin A2, but with many other annexins, and one is annexin A6, and we could also show that this is really the case. So annexin A6 is important because it's localized in the axonal hillock, which is actually the transition of the cell body of a neuron towards the axon. So probably this interaction is important to keep tau in the axonal compartment. And when we interfere with this interaction, we also saw, and we published it in this paper, that we lose the axon-specific localization of tau. So when we published these data, you can see here that tau interacts with annexin A2 via the exon 1, and tau binds to the core domain of annexin A2. Then we also presented this kind of a model. You can see here the microtubules and tau, the amino terminus of tau has a length of about 20 nanometer. Interaction occurs here with the extreme end terminus. So when tau is bound to microtubules, it can then link in a calcium-dependent manner to the plasma membrane. And by that, it keeps plasma membrane on distance. When we published that, actually, JCB, Journal of Cell Biology, titled "Annexin Keeps Tau on a Short Leash." So apparently that is one of the mechanisms of linking tau to the plasma membrane via an annexin-mediated mechanism. So that was the excursion why microorganisms are important also for our research in order to analyze, in this case, interaction of tau with other proteins. So I would like to move on with respect to the mechanisms of tau passes. So if you look at internet, and then you see lots of graphical representations what tau is doing and what's happening during disease. For example, this is a schematic from the NIH. It is shown here that tau binds to microtubules. It is mentioned here. It's stabilizing the microtubules. During disease, it gets hyperphosphorylated. Tau is falling off the microtubules. It's causing or it's forming these neurofibrillary tangles, and then the microtubules are disintegrating. So that sounds good. So tau stabilizes microtubules. Disease-like modifications like hyperphosphorylation or aggregation then interfere with this interaction. Tau detaches from the microtubules. Microtubules disassemble. Sounds too good to be true, and this is also the case because recently also it has been shown that animals can live entirely without tau. So tau KO mice have been produced, and they practically don't show any changes, although they don't show any changes with respect to microtubule stabilizing. So this mechanism, this idea, although it's prominently displayed, is apparently not correct, at least not in this simple form. There is actually another paradox. I call this here the pathomechanistic paradox. And the other paradox is a cell biological paradox. So as you can see here, tau decorates microtubules. The 12 times of tau, so the time, how long it interacts with microtubules, are considered to be relatively long in the range of seconds. Tau is highly concentrated in the axon, but also motor proteins like kinesins share the same binding site on the microtubules with tau. So you would expect that if you have a protein which is covering the microtubule surface relatively long, that it would have a strong effect on axonal transport. It would actually inhibit axonal transport. However, it has been nicely shown, mainly by the lab of Randy Nixon in the US, that neither overexpression or a lack of tau influence axonal transport, not only in vitro but also in animals. There is simply no change. So that means that actually if you go into the internet and see pictures like that, quickly forget them. They are not correct. So of course, you have to come up then with an alternative hypothesis. It's not just enough to destroy hypotheses which are existing in the field. So we tried to approach this question actually by looking not on a population of tau, but to look at the behavior of single molecules of tau. We performed super resolution microscopy, which allows nowadays with the respective techniques and also microscopes to analyze the movement of individual microtubules in a process in an axon of a neuron, for example. And we did that, and the approach which we were taking is we put a tag on the tau protein. The tag is a halo tag, and the halo tag allows then to label it with a fluorescence marker. So we choose to do it on the very amino terminus in order not to interfere with the microtubule binding region, which is here in the C-terminal half of the protein. So why don't we put directly a fluorescence marker on the protein? Well, the reason is that with this halotech approach, we can somehow tighten the amount of molecules which we are actually labeling. And we don't want to label a population, we just want to label very few tau molecules, which we can then follow over time. So it's in the range of every thousandth of tau molecule, which we can then label by that, but we can label it covalently. And then we perform a specific microscopy technique, which is called TIRF microscopy. And in TIRF microscopy, you are just producing a wave which irradiates a small portion of the cell which is close to the cover slip. So by that, we reduce the background from the rest from the cell, and we just concentrate on a very small region of the cell. We actually do it in the so-called halo mode, which is called dirty TIRF, because actually we are not only looking at the plasma membrane, but we're going a little bit deeper also in the axon with respect to the microtubule tau interaction. And with this approach, we can reach a spatial resolution which is lower than 20 nanometer, so which is much below the diffraction limit which you usually have with light microscopy, which is in the range of 200 nanometer. And we get, with a highly sensitive camera, temporal resolution which is in the range of 5 milliseconds. And we did that in cell nanos, and I was introducing that already before. And now I need your help actually in order to show how actually such an experiment looks. So this is a single tau molecule which is here interacting with microtubules in a process, in an axon-like processes, which is oriented like that. And we took images every 5 milliseconds, and you can see already that in this time which we are watching it, it is not binding to a particular region, but it's actually spreading over some distance, which is even more than 1 micrometer. So if we follow up on that, and when we visualize that over time, then you can see that in a relative short time interval, in a time interval of less than 3 seconds, tau shows many interaction, a single tau molecule shows many interaction in the microtubule array which you have in the axon-like compartment. So this was work which was done in a collaboration of a physicist, a PhD student in our lab, and a cell biologist, Maxime and Dennis. And based on this localization, we concluded that the interaction of tau on an interaction site, on a microtubule binding site, is very short, and then it hops towards the next position. And actually what we are visualizing with this approach is only the binding events. Besides the binding events, we cannot see tau because it's simply too fast. So what we see here is individual binding events on the microtubules, and from that behavior actually we concluded that it is highly dynamic and it shows a kiss and hop interaction. It shortly kisses a microtubule binding site and then it hops to the next position. So, and based on the single molecule data, we can also determine then the interaction time, the dwell time. And the dwell time is in the range of 40 milliseconds. So it's well below 1 second, much slower, much faster actually, as what has been reported previously. So from this experiment actually, we can see that tau interacts with microtubules by a kiss and hop mechanism, and the dwell time is in the millisecond range. When we started to do the experiments, I was very skeptical whether we could get additional information from single molecule tracking, because my opinion was biology works in a population of molecules. It not works via individual microtubules, or individual molecules, individual tau molecules. However, this feature of kiss and hop we would never been able to see when we simply look at the population, because then we see simply a distribution, a dynamic distribution, but we would not be able to determine the dwell time. Why do we think that this short dwell time is important? Well, for several reasons. Come to that in a second. But the first question actually which came up then was, if it's only interacting so short with microtubules, is it still able to modulate microtubule dynamics? And that was considered to be one of the role of tau protein. So we did a respective experiment. We did it using a fluorescence decay after photoactivation assay. What we did is we labeled tubulin with a fluorescent protein which can be photoactivated with a short laser flash. And then we can follow this photoactivated, this population of photoactivated molecules over time. So if we activate here a region in a process, so it starts to glow, to produce fluorescence, and then we follow the distribution of the fluorescence in this region over time, only in this region of activation, and what we see is then a fluorescence decay after photoactivation, or short FDAP. So the fluorescence is decreasing, but it's not due to photobleaching or any other effect, it's simply due to diffusion. Tubulin, a certain population, is diffusing out of this region of photoactivation. And based on this FDAP with the respective formulas here, which were again developed by Maxime, we can then determine how much of our fluorescent molecule is incorporated in microtubules. So, for example, if there's more incorporated, then the fluorescence decay would be slower, because less would be diffusing out. If we depolymerize the microtubules, and of course we checked that, for example with colchicine, then you would see a larger FDAP, then tubulin would diffuse out faster. This is actually the microscope which we are using. It's a laser scanning microscope incubated with an incubation chamber, which allows us here to do experiments at physiological conditions at 37 degrees. Here is a typical photoactivation experiment after expression of photoactivated tubulin. We perform here a photoactivation, then we follow the distribution of the signal over 112 seconds. We take an image every second. And then you can see here that it diffuses out of the region of photoactivation. And by that, then we can determine the amount of polymerized tubulin. Now comes the answer then to the question which I was phrasing in the beginning. So under control conditions, if we don't add any other protein, then you can see that about 2/3 of tubulin is actually incorporated in microtubules. That is also consistent with biochemical data on the incorporation or on the amount of polymer versus monomer or dimer. When we express the control protein, triple M cherry, that is our fluorescence marker, actually nothing changes. It's the same. However, when we express tau protein, coupled with M cherry, in order to be able to see that, then you can see that the amount of polymerized tubulin is significantly higher. It's more than 10% higher. So the question to the answer, which I was phrasing before, is actually yes. The interaction is still long enough, although it's very short, to promote microtubule polymerization, to induce incorporation of tubulin into axonal microtubules. So tau promotes axonal microtubule assembly despite its short dwell time. So why is that important? Well, according to the old model, the dwell time of tau is relatively long. It's in the range of seconds. The idea was that the interaction with microtubules is actually regulated via a phosphorylation-dephosphorylation cycle. So tau gets phosphorylated, it goes away from the microtubule surface, it's dephosphorylated, it comes back to the microtubule surface. Would be possible with a dwell time of 4 seconds. The kinases and phosphatases would actually be fast enough. However, you would expect actually that under these conditions, tau would be in the way, these black dots here, for efficient axonal transport. You would expect that there is an impairment of axonal transport, because if we calculate it, we would come to about 100 collisions per dwell time, and that would mean that there is a significant impact on transport. With our model, with the Kiss and Hop model, actually dwell time is in the range of 40 milliseconds. Microtubule regulation cannot occur any longer. No kinase phosphatase is that quick. So it is simply done via a dynamic interaction with the microtubule surface, via Kiss and Hop interaction due to a relatively low affinity. The result would that be that if we do the same calculation as before, that actually would come down to 1 collision per dwell time, and under these conditions we would expect that transport would not be affected too much. So the idea is that tau is interacting with microtubules in a highly dynamic manner, and by that transport of vesicles via motor proteins is actually not affected because tau is hopping away quickly enough so that the transport can occur here. So this is therefore a hypothesis which then shows that the binding of tau is long enough to regulate microtubule polymerization. That is what our FDAP experiments were saying, but short enough not to impair axonal transport. So the mechanism appears to be perfectly suited for a protein to regulate microtubule dynamics without interfering with axonal transport. So one consequence or one conclusion of such a behavior would be that if this interaction is changed in either direction, then a neuron might develop problems. So if the interaction would become longer, then you would expect that it would interfere with axonal transport, and that can then cause disease-related changes. So actually we checked whether that could happen, and what we found in the literature that tau actually can be cleaved by an enzyme, which is caspase 3, in a way that just the last 20 amino acids are cut off, so that tau becomes a little bit shorter. The microtubule binding region is still unaffected here. We did then experiments with deletion constructs where we successively deleted the C-terminus of tau. And what we found is then until we reach this red region here, actually the binding of tau to microtubules is increasing. The dwell time of microtubules is increasing. Therefore, we were wondering whether the effect of a caspase cleavage of tau, just getting off these last 20 amino acids, would actually also have such a consequence. We did single molecule tracking, as I introduced it before. Here you can actually see the trajectory of a tau molecule on the microtubule surface. And what we observed is that the dwell time of caspase-cleaved tau protein is significantly larger. It's binding longer to the microtubule surface than uncleaved tau protein, as I showed you before. And this is not only the case in model neurons, which we usually use in our lab, neuron-differentiated PC12 cells, but it is also present in primary neurons. Here we also see a significant increase in the dwell time. So the question would be, increased dwell time, would that interfere with transport? Short answer is yes. So if we determine the velocity of transport and the processivity, that means how often tau is going in the same direction or changes its direction, not tau but vesicles, or change their directions, actually both is significantly decreasing. So actually a longer dwell time leads to an impairment of axonal transport. Transport becomes less efficient because the velocity and the processivity is decreasing. So actually that was combined work by two PhD students in the lab, by Christian Conze and by Marina Riarola. And of course we were interested, could that be a mechanism which also is of relevance in disease? So what we checked is an increased cleavage of tau at the C3 site, also in Alzheimer's disease patients, specifically in the hippocampus. That is a region which is affected in the disease. Actually, we did check that with a Western blot approach, and you can see actually that this is exactly the case. So the tau C3 amount is actually increasing in AD patients compared to controls. But that is also associated with an increased amount of total tau protein. So actually it is not a specific increase of this cleavage, but in general the amount of tau is increasing, and since tau is cleaved to a certain percentage, also the cleaved tau is actually increasing. So does that have a consequence? We tested that in organotypic cultures of the hippocampus of a mouse. And we introduced then either wild-type tau or tau C3 with a virus system. We usually use SynPis virus, where we can put a pulse of tau expression in the neurons. And then after some days we determine the morphology of the neurons, and you can nicely see actually that the expression of tau C3 leads to a dendritic atrophy. So the dendrite becomes much more simple. It loses actually branches and it loses processes. So that would generate a problem for the neurons because the connectivity would be disturbed, would be actually decreased. So actually in a mouse model we could also show that tau C3 cleavage is actually increased during aging. So in contrast to this here, there is really an increase during aging. So perhaps that is a general aging process, that tau gets cleaved. The problem is then that this cleaved tau binds longer to microtubules, interferes with axonal transport, leading to dendritic atrophy, which is then taking place in addition to other changes in Alzheimer's disease patients. So, in the last part of my presentation, I would now move towards therapy. So, we could describe what probably is going wrong, but of course the main reason of doing scientific work is not just to monitor what is going wrong, but also to try to interfere with that and go in the direction of potential therapeutics. We are basic scientists, of course. We are far away from really developing drugs for the patients. However, in our opinion, it's very important to understand the mechanism of a disease in order to develop mechanism-based approaches, which then can target critical aspects so that neurons survive better and the patients develop dementia not only to a later time point. So, an important aspect which I didn't mention to you before is actually that we heard already that tau is interacting with microtubules via its microtubule binding region. However, the same region here which is interacting with microtubules is also involved in its aggregation. So that generates a problem. If we want to use drugs which inhibit tau aggregation, then a consequence could be that tau would also not be able any longer to bind to microtubules, which we want to avoid. So the aim of drug development, and we developed that some time ago in drugs, is that a tau targeting drug needs to inhibit tau aggregation, but at the same time it also needs to restore physiological microtubule interaction. There is also another aspect which I would also like to mention later. So the toxic species appears not to be the higher aggregates like these paired helical filaments, but soluble oligomers. So if we develop aggregation inhibitors, it would be probably more safe not to touch already the formed aggregates. Because if we cause a disassembly of these aggregates, then it could happen that by that we actually increase the amount of toxic species. So probably the cell is trying to remove toxic species by somehow making it biologically inert and having these higher aggregates. That is a little bit I think perhaps misleading. If you simply look at the brain and you see these aggregates and you think, oh, these aggregates are the problem. Does not need to be the case, it's only what you see. The problem can be one step further, like the soluble oligomers. So we want to develop actually a drug which inhibits tau aggregation, which restores physiological microtubule interaction, and which prevents the formation of tau aggregates, but better that does not touch existing tau aggregates. However, that is not easy, and in order to do that, you need sort of functional cell assays which are operating, and we thought that actually our assays are useful for that. So these single molecule tracking experiments are very informative. However, they are also extremely time consuming. So therefore we went back by knowing the data from the single molecule tracking experiment to actually develop an FDAP-based approach to determine in a population assay the interaction of tau with microtubules. So what we do is actually very similar to what I showed you before, but in this case we simply label tau with photoactivatable GFP. Then we can do the same. We can perform a photoactivation, and then we follow the decay of the fluorescence over time. With that, we see a decay curve. This here would actually be the experiment. It's a 6 micrometer region which we are photoactivating. And you can see here a decrease in the fluorescence, which again is due to the diffusion of tau out of this region of activation. So again, from the FDAP curve, we can learn how much tau is actually binding to microtubules. So if it would bind less to microtubules, then the decay would be faster. If it would bind more to microtubules, the decay would be absent or would be significantly slower. In addition, and that was again the work of Maxime, we could do some modeling approaches, because in a system like our model neurons, we know very well the distance between the microtubules. So from that we can also calculate the number of interaction sites, and this allows us then to do a modeling approach where we not only can see the effective diffusion constant here and calculate the effective diffusion constant, but we can actually also calculate the k-on and the k-off value of this interaction. And we see that actually with this modeling, and also for that the single molecule data were very important, it fits to actually the behavior of single tau molecules. So how do we go further? There are patients, there's an Alzheimer's disease patient and also a patient with another tauopathy, which have a mutation in tau protein, and the mutation is here in the microtubule binding region. It is a delta K 280 mutation, means that the lysine at position 280 is lacking, is missing. So it was published that this mutation leads to an increased aggregation of tau. So we thought that is a perfect model to check our system whether we can visualize actually a decreased microtubule interaction due to an increased aggregation of tau. So first, of course, we checked whether the literature is correct. Not everything which is published is correct. That is also something which you learn when you are a scientist for several decades. So we produced recombinant protein of either wild-type tau or the delta K mutant. We performed an in vitro aggregation assay, which works in the presence of heparin. That is a published procedure. And then we simply determine the amount of aggregated tau protein after ultracentrifugation. You can see that with wild-type tau, there is an increased aggregation over time. We are speaking here about 2 days in total. And when we take our delta K 280 mutation, then the aggregation is increased at all time points. So at least on the in vitro level, it's really an aggregation-prone tau mutant. And when then electron microscopy of the aggregates which are formed, then you can see they look very similar to the aggregates which have been isolated from patients, paired helical filaments. You can also see nicely here these regular turns. So this distance which you have between the turns. This was work which was done by Nicolo in the lab. So the next step then was to check what happens in a cellular context. So we expressed either tau, wild-type tau, or our aggregation-prone mutant in our model neurons. We we then perform our FDAP experiments. We perform the laser activation of PAGFP tau, and we follow the distribution of tau over time. So from that, we can determine an effective diffusion constant, and we can also determine K-on and K-off values. You can already see here directly from the raw data that compared to wild-type tau, the delta K construct here shows a much higher increased decay, showing that it interacts less with microtubules. So that is also reflected by the effective diffusion constant. It is much higher for the delta K construct, and by the calculation then of the microtubule bound tau, which is much lower. Therefore, the difference between wild type and delta K would then be actually the amount which is aggregating, which is forming oligomers. By that we have an indirect assay for tau aggregation. So tau delta K 280 shows impaired microtubule interaction in axon-like processes of model neurons. Now we can use that as a screening system. And we did that by, well, first actually we wanted to check whether that is really also correlated with tau aggregation. That is done by experiments from Ahmed, was coming from Egypt and did his PhD in our group. He prepared primary neurons from the dorsal root ganglion of mice. The advantage of this approach was that we can perform also a long-term expression of our constructs. And the idea was that perhaps after long-term expression we can really see higher aggregates, and we can correlate that with the microtubule binding in order to prove that it's really the aggregation which is causing then the decreased microtubule interaction. So he did the same experiment here in DRG neurons. In this case, they were transduced with a lentiviral for vector for long-term expression. Again, you can see an increased fluorescence decay of our delta K mutant, also an increased effective diffusion, decreased binding of tau. And when we look over time, then you can see here already after 7 days, which is then increasing up to 28 days, we see aggregates in the cell bodies. So we don't see it in the axon actually. That is very much reflecting what we also see in the disease, that the aggregates are forming in the cell body, which we can then visualize with AMI tracker, which is a marker for aggregates, for beta sheet aggregates like they are forming with tau. And you can nicely see here that together with an increased number of aggregates, we see also the decreased interaction with microtubules, confirming that really it's the aggregation which is interfering with interaction with microtubules. Or in other words, if we monitor microtubule interaction, then we can also monitor effects on aggregation. So we teamed up with 2 groups in Italy. Giulio Rastelli, he's actually a modeler, and Daniele Pasarella, he is a chemist. And Daniela was synthesizing a whole array of different 2-phenyloxazole derivatives, just call them FOX derivatives. Actually, when we wanted to publish this paper, we first called it POX, and then we thought, okay, if you want to publish a treatment, then POX perhaps is not a good name, so we renamed it then to FOX. Giulio actually was predicting that these 2-phenyloxazole derivatives might be actually a polypharmacological drug which would interfere on one side with kinases, but on the other side also with tau aggregation. However, that is just modeling. You have to confirm that in an experimental setting. And that's actually what we did here. We determined the effective diffusion constant. We analyzed then several of these FOX compounds. And you can see that one of these compounds is actually showing a largely decreased effective diffusion constant, which is a putative tau aggregation inhibitor. So you can see it here, that the effective diffusion constant is decreased with our aggregation-prone construct, suggesting that it's interfering with or preventing actually tau aggregation. So actually we can also do a dose response curve, and we see an IC50 in the range of 14 micromolar, and actually at high concentration the effective diffusion constant goes down, almost goes down to wild type tau. So this would suggest that it's able to completely restore actually physiological tau microtubule interaction to a physiological level. Did it also affect the aggregation of tau? We did it with our in vitro assay. You heard already that before. And actually, when we increase the concentration of FOX15 in this aggregation assays, then we see also a statistically significant decrease in aggregated tau, but not only in, thank you, but not only in aggregated tau, but also in wild-type tau. So it doesn't depend on this mutation. Also, wild-type tau is actually aggregation of wild-type tau is inhibited. Sorry. So that helps a lot. So we tested whether this aggregation inhibitor also acts on preformed aggregates. I told you before, actually we don't want that. We only want to inhibit de novo aggregation, but not dissolve existing aggregates. And actually the result is no, it does not. And it also does not with respect to paired helical filaments, which we have isolated from patients with Alzheimer's disease. Although their FOX15 is not reducing the amount of the aggregates. So FOX15 inhibits tau aggregation in vitro, but it does not affect existing tau filaments, and it's also restoring the physiological tau microtubule interaction, as you could see it before. So Giulio did some modeling, and according to the modeling, actually he found cryptic channel-like pockets crossing tau protofilaments, and actually he was also identifying that FOX15 disrupts the propensity of a glycine triad at pocket 2. That here is a pocket 2. That is here where we also have the interactions between protofilament A and B. And by that it decreases the propensity to form PHF-like assemblies. That would also be an explanation why it actually interferes with the de novo aggregation of tau, but it does not affect existing aggregates. So it's a nice confirmation also by a modeling approach of our experimental data. I told you before that FOX15 is actually a polypharmacological drug. It also binds to the ATP pocket of GSK3 beta based on docking analysis. GSK3 beta is a very important kinase because actually it's one of the kinases which is thought to play an important role in the hyperphosphorylation of tau. So that an increased activity of GSK3 beta then leads to the increased phosphorylation of tau, which then triggers perhaps further disease development. Actually, also in vitro assays, which were done by in the lab of Giulio, he could show that FOX15 really inhibits GSK3 beta with an IC50 of 1.9 micromolar. We were then testing whether it's also affecting tau phosphorylation in our cell system by doing a phosphoproteomic approach. So by that we compared then cells treated with carrier with cells which have been treated with a drug, with FOX15. We performed then a phosphopeptide enrichment analysis which allows to identify all phosphor sites in proteins in the cell. We concentrated then on the tau phosphor profile. And you can see here that in FOX15 treated cells, many of these phosphorylation sites, including many of these GSK3 beta sites, in this proline-rich region show a reduced phosphorylation, significantly reduced phosphorylation. which is interesting because this proline-rich region is considered to be the region which regulates the tau microtubule interaction. It probably not only regulates the tau microtubule interaction, but also the tau-tau interaction, means the tau aggregation. So FOX15 is working on one side as an aggregation inhibitor. On the other side, it is also decreasing the phosphorylation of tau at a potentially important site at the proline-rich region. So, the model, every talk ends sooner or later with a model, is therefore that tau, which is interacting with microtubules in a highly dynamic manner, gets phosphorylated. It also gets phosphorylated under physiological conditions. Probably the phosphorylation is increased under pathological conditions. This phosphorylation then leads to the formation of monomers and soluble oligomers, which are probably the toxic species, which then leads to the formation of paired helical filaments and later to neurofibrillary tangles. So our drug candidate, FOX15, inhibits the formation of protofilaments and PHFs, as we could show it in vitro, and it actually decreases the phosphorylation. It decreases the oligomers, and it rescues the physiological tau microtubule interactions, so that tau is able to dynamically interact with microtubules as it would naturally be doing. So very recently we also teamed up with another group, with a group of Sandrine Angerie in Paris, together with Niccolo Tonali, who is also leading a group there. In order to use our assay to test whether also other compounds might be active. And actually their idea, I found quite clever, was to start from a natural compound, HSP90. It's a heat shock protein, and the heat shock protein is known to inhibit aggregation of proteins in a cellular compartment, in the cell at all, in order to allow it to function properly. So the idea was that to take the regions of HSP90, which are involved in preventing aggregation, and make peptides out of that, make modified peptides out of that, which are not degraded any longer, but are taken up by cells. And we tested one of these compounds, which Sandrine called beta HSP90, and you can see that this compound is also active in reducing the tau aggregation by decreasing the effective diffusion, by that increasing the interaction with microtubules, and it actually works mainly on the kaon level, so that tau is actually then better binding to the microtubules. So these are the experiments with the delta K 280 construct, the aggregation-prone construct. When the same experiment was done with wild-type tau, there was no effect any longer. So that would suggest that actually we do not have off-target effects, but the drugs, these peptides, are very specifically reducing tau aggregation without touching the physiological tau microtubule interaction, are being able to restore physiological tau microtubule interaction. So with that, I'm at the end of my presentation. I hope I didn't bore you too much. I would like simply to sum up what I tried to convey to you, is that we showed by a single molecule analysis that tau shows a high dynamic interaction with microtubules, a kiss and hop mechanism, and that this high dynamic interaction makes sure that it's not in the way of axonal transport, but still able to regulate microtubule polymerization. I also showed you, with respect to the caspase 3 cleavage, that disruption of the dynamics of this physiological tau microtubule interaction can cause problems. It leads to an increased dwell time binding to microtubules, which then inhibits transport, which is vital for neurons. So the result is that a dendritic atrophy is occurring. I hopefully could convince you that this quantitative live cell imaging approach is not only useful to answer basic scientific questions, but it's also a good tool to screen for tau aggregation inhibitors that restore physiological microtubule interaction, and this aspect is important because aggregation and interaction is done by the same domain, by the microtubule binding region. And I could show you that two of the compounds are quite promising. They are now a starting point for a further improvement. And these are FOX15 and beta HSP90, which inhibit tau aggregation and restore physiological microtubule regulation in neurons. Perhaps one remark, I think I forgot it to mention before. FOX15 is a polypharmacological drug. It not only affects tau aggregation, but it also affects kinases, also the famous tau kinase GSK3 beta. Beta HSP90 is some sort of also polypharmacological because it inhibits aggregation in general. And in this paper, which is now actually in press in Nature Communication, should come out in the next days, we showed that beta HSP90 is not only inhibiting tau aggregation, but it's also inhibiting A-beta aggregation. Therefore, it actually works on both key proteins, can be also considered, therefore, as a polypharmacological drug, and hopefully be of help in treating this devastating disease. So with that, I would like to mention the people who did the work. Our department is led by two PIs, that is Lydia and myself. These persons here, I was introducing them through my presentation. They left already the lab. This is the current crew in our lab. We are very happy with them. We also have two Indian PhD students in our group, Lisha and Nirmal, together with Anna from Iran, Larissa from Brasilia, Miltos from Greece. So we are a quite international group. Actually, the only nationality which is missing among the PhD students is German. We collaborate with cell nanos. I was introducing that before with high-end microscopy. Very helpful is here Rainer Korre and Jakob Pieler, who is heading this cell nanos unit. We work also with human brain samples, which come from the University of Leipzig, from Thomas and Max. And I introduced some of the work with yeast experiments with Jürgen. Actually, what I did not mention is also important, that we also use the yeast system to generate viruses, because yeast shows high recombination activity, and we use yeast recombination in order to make efficiently viruses, because usually they don't have too much cleavage sites in order to do classical molecular biology. So it's extremely helpful. With respect to tau aggregation inhibitors, we collaborate with Giulio and Daniele, and also with Sandrine and Nicolo. So I hope I didn't forget anybody. You can see here our lab, as it was some time ago. We were visiting a SEAL station here at the North Sea. We are relatively close to the sea, so that is what we do doing lab excursions. For example, I should also mention that part of this study was also financed by an EU, which is called Turbine Train, where biologists and chemists were working together And I think that is quite a useful combination, as hopefully you could also see during my presentation. So with that, I'm at the end. Thank you very much for your attention.
Any questions?
I can't hear you very well.
Sairam, I have a couple of questions. One, the FOX15, it's a phosphorylation process, right? When you use FOX15, it phosphorylates or inhibits phosphorylation.
It inhibits phosphorylation by working actually on GSK3 beta, but we also have evidence that also it inhibits phosphorylation of CDK5.
Yeah, CDK5. So I want to ask you, what are the side effects in general, systemic side effects, number one.
Yeah.
Second thing is, have you ever tried immunotherapy using monoclonal antibodies? So that they can bind to the aggregates, and then the macrophages come and eat them up. So,
yeah. First question about side effects, we don't know. Of course, that is a disadvantage of polypharmacological drugs, that there can be side effects, but that would then be the next step. As I said, our approach is to develop mechanism-based drugs, and this is actually a drug which addresses the mechanism which restores physiological tau-microtubule interaction. Of course, the next step would be, after we optimize these compounds, to do first studies in animals in order to see potential side effects. That may be a problem. Therefore, we try to make an array of different FOX derivatives which have differential effect on tau aggregation and kinases, because that might also be an aspect which is important, just to titrate out how this polypharmacological effect would be. So, with respect to immunotherapy, I was mentioning in the beginning that the first monoclonal antibodies are on the market. These monoclonal antibodies are targeting A-beta. The problem with these antibodies is, first, they are very expensive. Second, they produce side effects like brain swellings. So they are not allowed to be used in patients which have a genetic predisposition for Alzheimer's disease, for example, with respect to the ApoE4 allele. So, and they should be used very early in the disease process, otherwise they would not help. So it's an interesting approach because it is targeting not just the symptoms, but the cause of the disease. But according to my view, that is just the starting point. There need to be improvement, and antibody therapies have lots of side effects either. It is different whether you use them for cancer therapy, like they are used, because their side effects can be borne better than with neurodegenerative diseases for the elderly. And actually, with respect to tau, that would probably be not a good way, because tau is an intracellular target. So A-beta is extracellular. So if you get an antibody into the brain, then you have a chance to affect A-beta aggregation. To get an antibody into a cell, that is another issue. Therefore, with respect to tau, I think that is probably not an approach which would work. There are trials, but the concentration of the antibody is extremely high, The side effects are extremely high, so I doubt that it will ever work. I think think more peptides or small molecules will be more helpful in that respect. Can you?
Single chain fragment variables, which are hardly 28,000 molecular weight. So they are very small.
Yeah, the llama antibodies, for example, yeah. In principle, these nanobodies could be useful, could be more useful actually with respect to A beta treatment, but I still am not sure whether it would really work good for an intracellular target.
What about cytokine therapies? Cytokines.
Cytokines. Inflammation is occurring during disease, and targeting inflammation is always a good approach. So during neurodegeneration, then tau is also released, also that causes immune reaction. So that is another avenue of research. That would not be a causal therapy. I'm still convinced that A beta and tau are the causal part of the disease, and for me, therefore, the prime target. But in order to symptomatically treat patients, I think it's a very good approach also to model or modulate the immune reactions. Mm-hmm.
For paucity of time, we'll stop for questions. People can discuss with sir during lunch break personally. Thank you.
Okay.
Thank you, Professor Roland.
Professor Roland Brandt's pioneering research on how neurons carry out their vital functions and how their failure leads to disorders like Alzheimer's and even shape the way the world understands neurodegeneration. For us, as students and researchers, his talk is not just knowledge, it is inspiration, a chance to learn from a scientist who has turned curiosity into discovery. To honor him, I now invite Professor S. Venkatesh, Head of Department Biosciences, and Dr. A. Ashok, Department of Biosciences, to kindly come forward and felicitate him. Sairam.
Om Shri Sai Ram. Now, I have the privilege of introducing next speaker, Dr. Sridhar Narayan. Dr. Sridhar Narayan, a passionate drug hunter with more than 20 years of drug discovery and developmental experience in Indian pharmaceutical industry in various therapeutic areas. Dr. Sridhar holds a basic degree in pharmaceutical sciences from University of Mumbai, a PhD in pharmacology from the Ohio State University, and has postdoctoral experience in neuropharmacology at the University of California, Los Angeles. Dr. Sridhar, a serial entrepreneur, is currently founder, director, chairman, and chief executive officer of Foundation for Neglected Disease Research, FNDR, a not-for-profit company established in 2014 with a mission to discover and develop drugs for diseases in the developing world. Dr. Sridhar is also the co-founder of Peptris Technologies Private Limited, an AI machine learning company working towards the discovery and development of new drugs in rare diseases, oncology, and inflammation. Prior to this, he was appointed Vice President and Head of Innovative Sciences for the Infection Innovative Medicines Group at AstraZeneca, India, and was responsible for the discovery and development of potential clinical candidates in the TB and malaria. Throughout his career, Dr. Sridhar has overseen the discovery and development of one drug, enmetazobactam TBA 7371, ZY19489, and 15 other clinical candidates in the areas of infection, oncology, diabetes, inflammation, and respiratory diseases. He has also executed out licensing deals with major pharma as well as in-licensing of candidates, NCE, NBE, which are in active development. This has generated upfront revenues in excess of 180 million USD. As part of FNDR, Dr. Sridhar has managed to raise donations worth 2.7 million USD from AstraZeneca, more than 2 million USD in grants money from funding agencies, and around 30 million USD in investment into preclinical and clinical assets development by partners over the last 8 years. Dr. Sridhar also serves as the Senior Technical Advisor at the Indian Pharmaceutical Alliance, in role in which he is actively involved with developing an innovation ecosystem at government, public, and private levels. Dr. Sridhar is a mentor to several startups, members of advisory boards, joint research community, academic and industrial collaborations, and served as a PhD guide. He has more than 50 publications and 25 patents to his credit. Dr. Sridhar and his organizations, FNDR and Peptris, have been the recipient of several awards at the national and international levels, including the Jack Beale Award of Outstanding Alumnus, a Lifetime Achievement Award from Indian Society of Clinical Research, the Longitude Prize Award, and Smart Bio Award, to name a few. Let's invite Dr. Sridhar for his talk. Thank you.
I'm not sure I need this. Very good afternoon, and first of all, thanks to Pradeep and Dr. Balram for inviting me to be part of this session. It's going to be a tough ask following Professor Roland. I think a phenomenal lecture on basic science, and it took me back to my days as a graduate student where I worked on neurosciences. So thank you very much for that, Professor. Now, generally, when I give a talk, especially to students, I like to make it very interactive. Yeah? One of the reasons is, as a student, I used to fall asleep whenever there was a session like this, right? So I want to make sure that you don't fall asleep. So I will ask you a few questions, make sure that you are awake, keep it interactive as such. Let me see. So I want to talk to you today about a disease called tuberculosis, right? How many of you have heard of it? Everybody's heard of it, right? How many of you know somebody who's had tuberculosis? Very few of you. And the reason being, it is considered a big stigma today, right? Even today, tuberculosis is considered a stigma. If somebody has cancer, they would go and tell somebody else that I have cancer. How many people will go and say I have tuberculosis? Very few of them, right? So the stigma that is associated with the disease is one of the reasons why there is not much of an awareness that is created. What I'd like to do today is to bring about a little bit of awareness of tuberculosis, a little bit of knowledge about how drug discovery and development happens, and possibly at the end of the lecture, some of you would understand what drug discovery is and how a drug is actually discovered. I'm going to take off from where Professor left in terms of basic science and take you through a journey where you will understand how a pharma company works, how drugs are discovered, and all in the drug discovery pathway. So, like my friend here mentioned during the introduction, I started this organization in 2014 with the primary view that it would work on diseases which have a very high social impact. And we wanted to focus on infectious diseases because they are the major cause of death in India even today. And most of them can actually be prevented or treated very effectively with drugs. So the areas of focus for my organization, we work on mycobacterial diseases. Can I have the pointer, please? Okay. Yeah, thank you. So we work on mycobacterial diseases. We work on mosquito-borne diseases, a lot of gram-positive, gram-negative, and fungal infections, and also zoonotic diseases like rabies. The organization is about 15 ongoing projects across drugs and diagnostics. We have 2 drugs in clinical development, a number of patents that have been granted, several publications, and about 37 employees today as a strength. This ESL3 facility that we have based out of Bangalore, mainly we are able to do biosafety level 3, biosafety level 2 work, a lot of the microbiology work, virology, rabies, cell culture, biochemistry, drug metabolism, pharmacokinetics, bioanalysis, and also our own medicinal chemistry. So in essence, we are a self-sufficient unit which is capable of doing end-to-end preclinical drug discovery. This is a photograph of our biosafety level 3 facility. So you can see a very nice biosafety cabinet. We have an animal system which is individually ventilated cage system. So you can have multiple animals with different infections housed within that. And anybody in safety level 3 has to use something which is a astronaut-like hood that we are having, and each of these is connected to a HEPA filter. So the air that comes into the person's face is fully HEPA filtered air, so there's a very minimal chance of infection that the person gets. So these are the kind of working conditions that we provide to our employees. Like he mentioned, I want to also acknowledge my co-founder, Dr. Radhakrishnan Shandil, who was with me in AstraZeneca, and together we started this company. We started this with an initial donation of $2.7 million from AstraZeneca, and that enabled us to build the organization. But 11 years into the organization, we are absolutely self-sufficient. We have no dependence on AstraZeneca as a unit, and everything that is managed within the organization is fully generated through funds which are generated by the work that we do within the organization. We like to work in partnership. Being a not-for-profit, we work extensively in partnership. We have multiple collaborations, contract research work, licensing deals that we do. variety of preclinical animal models that we are involved in. And to date, we have more than 100 clients and collaborators from across the world. So I'll touch a little bit upon the drug discovery pipeline that we have within the organization. So if you look at drug discovery itself, you have multiple areas starting where you have the identification of a chemical molecule. So like Sir mentioned, FOX15 would be your ideal hit candidate, right? Where you see the first glimpses of biological activity. And from there, it is about a 10 to 12 year journey for the molecule to become a drug, right? So this entire process from hit to lead optimization, preclinical studies, and the clinical trials itself would take anywhere about 10 to 12 years, depending on what therapeutic indication you are working on. If it is something like tuberculosis, that can go even up to 15 to 20 years, right? Our pipeline, we have multiple candidates, 2 which are in phase 2. I will be talking a little bit about TBA 7371, which is our best candidate so far. Chloroquine, most of you are familiar with. This is an anti-malarial drug, which we are repurposing for the treatment of tuberculosis. We have multiple others in the pipeline, and I'll touch a little bit upon this repurposed drug for the treatment of rabies. In addition to the drugs pipeline, we also have a significant pipeline when it comes to the diagnostics and devices. We are working on developing an LCMS-based diagnostic for the diagnosis of both pulmonary as well as extrapulmonary tuberculosis. We differentiate between bacterial and viral infections, lymphatic filariasis diagnostic, and a device which depletes antibiotics from wastewater, which allows for us to provide for clean water that is coming out of hospitals, out of municipal ETPs, and also out of pharmaceutical industries, which send out a lot of the water into the environment. So our device actually absorbs a variety of these and makes them into clean water. So now coming to tuberculosis, right? So tuberculosis is caused by Mycobacterium tuberculosis, which is one of the most successful microorganisms in the world. And the reason I call it very successful is because it has killed more people than any war or any other kind of disease, right? And it has been known, I would say, even almost 4,000 years BC, right? Even in the age of pyramids, there was existence of tuberculosis. So if you look at the disease itself, it's been very successful. It is found across the globe, right? So there are cases in the east, there are cases in the west, and now with the use of many immunomodulatory drugs, you're seeing a significant increase in something called as extrapulmonary TB, which I will touch upon a little bit, right? Host immune system dictates the disease. Annually, we have about close to 11 million cases of tuberculosis every year, right? With 1.5 million deaths per year globally. 90% of which happens in the developing world, which is the global south. 50% of this in India and China alone, right? We have about 2.67 million cases which have been reported in 2024, right? The government wants to eradicate TB or eliminate TB from India by the year 2030, right? How many of you think this is possible? Can I have a raise of hands? How many of you think 2030 is achievable? I have one gentleman there who's very optimistic. Excellent, right? So yeah, I'm sure that, we will get somewhere close to the goal. I don't think we will eliminate TB by 2030, but at least we should be able to, with all the new drugs that are coming up, be able to manage TB to a certain extent. In India alone, TB killed about 3.3 lakh people last year. Out of all the cases of TB, 2.5% of them are reported as drug-resistant tuberculosis, right? Which means they are either resistant to one TB drug, 2 TB drugs, or in cases of MDR, you have multiple drug resistance. So it is resistant to a variety of drugs that are being used for therapy. And there are cases of XDR today and TDR, especially in hospitals in Bombay, which have been seen, which means they are totally drug resistant. That means you will die of tuberculosis because there is no treatment that is available because of the drug resistance that comes up, right? And it's not a fast death sentence. It will go on for years, and it will be a slow and painful death that happens because of tuberculosis. So what is so special about this organism, and why is it so interesting, right? Most of you have looked at gram-negative infections. How many of you have taken an antibiotic course? Raise of hands. Almost everybody, right? Everybody's taken an antibiotic course. How many of you have completed an antibiotic course? You got 50% of them saying, yes, we have completed an antibiotic course. And how long is the course? Maybe 3 days, 5 days, 7 days, 14 days at the most, right? Now with tuberculosis, the challenge is the drug treatment goes on for months together, and if you are a drug-resistant tuberculosis patient, it will go on for years together. I'll touch upon those as we go along. So why is this a very intelligent organism? It can exist in multiple states. So you have an active TB, and dormant TB is something which is seen in almost 30 to 40% of the Indian population, right? So in this room of about a few hundred, I would think that there would be at least a handful, if not more, who have latent TB, right? And it affects all strata of society. It's not restricted only to the marginalized, right? And today, if you look at the way society is, all of us have house help. We never test them for TB, and the people who we expose them to are our children and our senior citizens. So the chances of TB getting to the most vulnerable populations is very high. So the TB exists in a variety of physical states. You have an actively replicating TB, a non-replicating TB. TB can be both intracellular as well as extracellular, right? And exists within granulomas and also within fibrotic lesions, right? So compare this to a systemic infection with a gram-negative organism. This is totally a hidden menace, right? So it's very difficult to treat tuberculosis because the drug has to cross multiple layers and also be effective against multiple states of the organism. So the need is to have bactericidal therapeutic targets which can actually work on multiple of these states of tuberculosis, whether they are replicating or whether they are non-replicating, right? The normal TB regimen today, I'll touch a little bit upon extrapulmonary TB also, right? So this is something which is significantly increasing and is increasing especially in countries like India, where there are several biosimilars which are being introduced into the market for the treatment of various diseases, right? So most of these biosimilars have an immune component attached to it, right? You will have some degree of immunosuppression which happens, especially with treatments for arthritis or cancer. And when you have this kind of immune suppression or an immune compromised nature, the TB bacilli start growing up again. So the latent TB becomes a full-blown TB. And a lot of this full-blown TB doesn't happen as pulmonary TB, it happens as extrapulmonary TB. So when you look at the different sites that are affected by the TB bacilli, right? You could have anywhere from the brain, the meninges, to the spine, to the pleural cavity, to the abdomen, your genitourinary tract, bone, and also the, right? So the TB bacilli can sit in any of these places and actually get reactivated in cases where there is an immune compromised nature. 4 TB patients is today an EPTB patient, right? So that means 25% of all reported TB cases are extrapulmonary TB, of which 30% are pediatric TB cases. And there are absolutely no drugs which are available for the treatment of EPTB in pediatrics specifically, right? The general regimen of rifampicin, isoniazid is used even there, yeah? And around the world, there are about 1.3 million cases of extrapulmonary TB. So what is the drug regimen for tuberculosis, right? So you have the normal drug-sensitive TB. You have a 4-drug regimen, which is 2 months of isoniazid, rifampicin, ethambutol, and parazinamide being given, followed by 4 months of isoniazid and rifampicin, right? So this is a 6 month of therapy. If you go to a resistant TB, there is a newer version that is available, which is bedaquiline, pretomanid, and linezolid, and is for 6 months, right? Whereas if you go with the standard regimen for drug-resistant TB, it can go anywhere up to 18 months of therapy, yeah? So the challenge is long treatments, low adherence, drug resistance, and a very high failure rate. So if you look at the standard of care for TB therapy, you have the short course, which was introduced in 1970, and that is still the standard of care today, right? So after almost 50 years, we are still existing with the same therapy that was used for the treatment of tuberculosis, which is HRZ for 2 months and the HR for 4 months. And with this therapy, after 2 months, you will see almost a 90% reduction in the TB bacilli, right? And it is human tendency. Once you start feeling better, what do? Stop taking the medicine, yeah? So after 2 months, if you stop taking the medicine, you have a relapse chance of about 70%. And this relapse most likely will be a drug-resistant tuberculosis, right? For people who continue this for 4 months, the relapse rate is about 40%. And even the treatment and complete the treatment, you still have a relapse rate of about 6%, yeah? So people who are on therapy for drug-sensitive tuberculosis will move towards being a drug-resistant tuberculosis, A, if they don't complete the course, and B, even if they complete the course, there's a small possibility that they will actually turn into a drug-resistant tuberculosis. So the drug-resistant tuberculosis, mainly because of AMR, which results due to the lack of completion of therapy, you will have about significant numbers of about 0.4 million people which are affected by MDR-TB. And in most of these cases, the success rate of treatment is very small. It is improving with the newer regimens, but the success rate was as low as 30% 10 years ago. Today it is about 60%, but we are not still at 100%, right? So the goal is to discover drugs which will get you to that 100% mark. In fact, the tuberculosis, drug-resistant, rifampicin-resistant tuberculosis is classified as a critical WHO pathogen in 2024. It was removed from the list earlier. It has been brought back because the number of TB cases are either stagnant in multiple countries or are going up. They are not showing signs of coming down. So for drug-resistant TB, right, the number of pills that the person takes in India for the treatment of DR-TB, you're looking at about 15,000 tablets being taken by an individual for the treatment of drug-resistant TB, right? When we take 10 tablets, we are tired of taking medicines. These people have to take 15,000 tablets with serious side effects and accompanied by at least 240 injections of amikacin, right? Or one of the amikacin class of drugs. Imagine that. And that is why most people don't complete the therapy, and you have a significant increase in drug-resistant tuberculosis coming up. So this is the newest regimen that is there. It is called the BPAL regimen, which is bedaquiline, pretomanid, and linezolid. And based on this had a 95% success rate in treating drug-resistant tuberculosis. This is just getting approved in India now, and hopefully this is available to the masses soon enough in India, which will make a huge impact on how TB is treated in India, especially the drug-resistant kind. So if you look at what we want as a new drug target product profile. So whenever we do a new project on drug discovery, the first thing we do is put out something called as a target product profile, which is the end goal that we want to reach when we discover a drug, right? So what we want as a target product profile is to have a drug which will treat MDR-TB, which will treat XDR-TB also. It will shorten the treatment duration, it will lower the dosing frequency, reduce the pill burden, and also be devoid of drug-drug interactions. Most TB drugs have some kind of cytochrome P450 kind of involvement. Rifampicin, notorious for the CYP3A4 related metabolism, right? So drugs which are metabolized through this will show a differential effect when given with the TB drugs. So which is why you want to be very careful when you proceed with development. You want to make sure that there are cytochrome P450 interactions are minimal. So this is the global pipeline of new TB drugs as of 2024. And from India, we are the only organization which has a presence on this pipeline, right? So we have one molecule here, which is FNDR 20364. Now this is a molecule that came from the soil from Antarctica. We had a project with the National Center for Polar and Ocean Research and Pyramids. This molecule was discovered from the soil, from a Streptomyces species, from the soil from Antarctica, and we've developed that further. And this works very well on both drug-sensitive as well as drug-resistant tuberculosis. And the other molecule which we talk about today is TBA 7371, which is a molecule which is a small molecule chemical entity that was discovered in Bangalore, developed in Bangalore, was funded by TB Alliance for clinical trials, and now is back in India for a further development. So when we started the project for TBA 7371, this was the target product profile that we put. Basically, we wanted an orally administered agent for the treatment of TB, which had efficacy and sterilizing effect in combination, appropriate dose for the combination, which targets both drug-sensitive as well as drug-resistant tuberculosis, a novel mechanism of action which would allow it to work in the drug-resistant strains, and no antagonism when given in combination. And the patient population that we were looking at was both drug-sensitive as well as drug-resistant tuberculosis. So this is the chemical structure of the molecule for those who are medicinal chemists in the audience, right? And you understand this belongs to the aza-indole class of molecules, the aza-indole being this group right here. And we had added a variety of substitutions. There were almost close to about 300 compounds that were made in this chemical class before we identified this molecule as the drug candidate for future development. So in brief, what this molecule does is a novel mechanism of action. It inhibits an arabinogalactan pathway in the mycobacterial cell wall, works on drug-sensitive, drug-resistant strains of the bacteria, and also is efficacious in the mouse model. I'll show you some data on that. A variety of the drug development effort gets stalled because of the regulatory toxicology studies, which start making it more and more expensive, right? And especially for companies in India, it's very difficult to invest in some of those because these can go upwards of a few $1 million in terms of the cost of doing the work. Then obviously the phase 1 and the phase 2 early bactericidal activity for the treatment of tuberculosis in TB patients. So I'll start with the biochemistry of this molecule. Basically, it inhibits something called as DPRE1, a key enzyme which is for the mycobacterial cell wall synthesis, and which is involved in the arabinin biosynthesis. Now when you inhibit this, the cell wall is not formed, and that results in the killing of the TB bacterium. In vitro, this has an MIC90 of about 0.64 micrograms per ml, so it's a very potent molecule. We have done an MIC in about 96 clinical isolates, which is a mix of drug-sensitive and drug-resistant isolates. And the MICs there were in the range of 0.04 to about 5 micrograms per ml. And if you look at its activity against the drug-resistant strains, which is here, you have significant activity against all of the resistant strains, whether it was resistant to streptomycin, isoniazid, or rifampicin. We've also looked at other DPRE1 inhibitors which are in development. One of them is BTC043, and our molecule works, though it works on the same target, it works in a mechanism which is different from that of BTC043. BTC043 is a covalent binder, whereas our molecule is a non-covalent binder. And even in cases where BTC is resistant, our molecule is effective. So if you look at this one, BTC is resistant here, but our molecule works. Again, these are mutations of the DPRE1 where the BTC-043 is ineffective and our molecule seems to work. So though it works on the same mechanism, it works on a site which is different from that of BTC-043, and that's why it is working there. We've also looked at translation from in vitro to in vivo. So you have a biochemical assay where the PIC50 was in nanomolar range, the MIC, which is about less than 1 microgram. We've done kill kinetics for this molecule. You will see AZ codes because this was discovered during the AstraZeneca days, and then it came back to the foundation. So you see the kill kinetics, you can see that it is both time dependent as well as concentration dependent. So the effect of the drug is dependent on both the concentration as well as the duration of treatment. We also looked at the oral PK, and the oral PK you can see very good levels in excess of about 10 to micrograms in terms of Cmax, and this is above what is required for the MIC. So a significant exposure in terms of time over MIC and also concentration over MIC, because of which you have significant activity of this molecule that is seen. And this is the animal model data. I'll go in detail on this one. So if you look at the animal model data, this is the animal model development. You infect the lungs with about 100 bacilli, and over a period of time of 31 days, you see a significant increase in the lung CFU of this bacilli. If you treat with rifampicin, you have a significant reduction in the lung CFU, and we see a very nice dose response with our drug at 50, 100, 200, and 400 milligrams per kilogram. So a very nice dose-dependent effect of our molecule to show that this is effective for the treatment of tuberculosis. We've also done a chronic model. So in the chronic model, you develop the infection, leave it for 28 days, and then you start the treatment, so which is a much more closer to what you would see in the human situation. And even in this model, you see a significant effect, the chromosomal aberration test, going to a 1 month long study in dogs and in rats, right? So you have a variety of studies that are done as part of the GLP toxicology studies. And all of these are done under something called as a good laboratory practice conditions. And most of you are familiar with what GLP is, right? So you will do these studies under GLP, and once it passes these studies and is found to be safe, you would then be able to get into the human trials. So we looked at a variety of studies. So you had a 1 month GLP tox in rats and monkeys in this case, where the no observed adverse event level was identified in both the studies. In the rat study, it was about 300 milligram per kilogram, and where the Cmax and the AUC was about 8-fold above what we expected to be the clinical exposure. In monkeys, also the NOEL was about 300 milligram per kg, and the NOEL was about 1.5-fold above the clinical exposure that we expected. In terms of the safety pharmacology, it had weak inhibitory effect against the phosphodiesterase isoform PDE6 specifically, and the PDE6 is involved with something in the eye, so this was something that we had to monitor as part of our clinical development package. There were absolutely no effects on the brain or on the respiratory system. Genetical toxicology studies were negative. There was no cardiac liability, no phototox liability, and no major red flags on repeat dose toxicity. So all in all of this indicated that this was a good candidate to move forward. So when we did a project assessment at the end of the GLP talks, you had almost 3 and 2 yellows. The yellows were one which were signals which we wanted to monitor in the clinic. All the greens went that the molecule was good enough to move forward into clinical development. So based on all the preclinical data, we wanted to establish what would be the starting dose that we should use in the clinical trial. So we did something called as a PK/PD modeling, which is pharmacokinetic pharmacodynamic modeling, which means you correlate the exposures to the biological effect. And based on this, you had the MIC of about 0.55 micrograms per liter, and you had significant exposures at drug given at 250 mg once daily and 500 once daily. And you could see that at both these doses, the plasma concentrations were significantly above what was required for the biological activity. So based on this, we predicted that there would be a human dose of anywhere between 100 to 200 milligrams as an absolute dose. So the part 1 of our clinical trial, so you do this in healthy volunteers, which is a single ascending dose. So in the single ascending dose, we started with doses of 100, 200, 400, and 800. You had 6 volunteers who were given the active drug, 2 who were given the placebo, a total of 32 subjects. We also looked at fasted versus fed to see if there was an effect of food on the level of the drug, and a variety of other cohorts which led to a total of about 82 subjects being part of your phase 1 trial. So this is data from the phase 1 single ascending dose and the multiple ascending dose. So you have 100 mg per kg giving you a Cmax of about 0.5 micrograms per ml, 200 giving an exposure of 1.5 and a AUC of about 87 microgram hour per ml. So based on our predictions, we had said somewhere in the range of 100 to 200, and 200 seemed to pan out very well for this, and this was the dose that we went into the clinic with for the efficacy study as well. This is the data from the 200 milligrams given repeatedly for 14 days, just to show that there is absolutely no effect of repeated dosing. The cytochrome related effects are not in any way altering the pharmacokinetics of the drug. So based on all of this data, we concluded that 100 to 200 milligram absolute dose is what we would go into the patient with. So this study was done in South Africa with about 90 tuberculosis patients who are drug sensitive. This was done at multiple sites, the Lung Institute in Cape Town, the TASC Clinical Institute in Cape Town, the perinatal HIV unit in Matlosana, and also the Aurum Institute in Schwane. So based on these 4 sites, a phase 2A dose escalation controlled randomized study to evaluate the early bactericidal activity was done in rifampicin sensitive pulmonary TB patients. The clinical trial design was like this. So you had the first cohort of 100 milligrams once daily, where there were 15 patients who were on the drug, TBA 7371, and there were 3 which were on the standard of care, which is HRZE. After this, there was a dose escalation that was done to 200 once daily, 100 mg twice daily. So you had a matching understanding of whether 100 mg twice daily was better than the 200 mg given once daily. You had 100 mg given 3 times a day, and also a 400 mg given once a day. So based on this, the clinical trial was conducted, and because of COVID, this trial took almost 4 years to complete, just for recruiting of 90 patients. The different parameters that we assessed between day -2 to day 14 was bactericidal activity, which was change in the CFU, change in the time to positivity, change in sputum LAM, and also some of the exploratory stuff in terms of the MIC shift. We looked at a variety of safety parameters, including the eye symptoms, which was a signal which came out from our GLP tox studies, and also a variety of PK-related parameters that were collected. So I'll just show you a snapshot of the clinical data, and the snapshot, what I want to look at is this line, which is the HREZ, which is the 4-drug standard of care, and the brown line here, which is our drug given at 100 milligrams thrice daily. So if you look at the CFU reduction that is seen with our drug alone, it is a 2 log reduction in CFU, whereas with HRZE, it is about a 2.5 log reduction. So the standard of care does about 2.5 log reduction of CFU, whereas our drug given alone does a 2 log reduction. So imagine what it could do when it is given in combination with the standard of care. So we expect that this would be a much effective drug when it comes in combination with some of the other existing drugs. So in summary, what we've identified is that we have a molecule which is very potent on the TB bacilli, which has good oral bioavailability, which is safe to use, and has shown efficacy in the human TB patient. Now, based on all of this, we are developing a sustained oral release formulation, which will be long-acting, either as an oral or as a long-acting injectable, which can be given once a month. So this is work in progress now. What we want to do next with this is bring this molecule back to India and do a long clinical trial in India with at least about 300 to 500 who are drug-sensitive tuberculosis as well as drug-resistant tuberculosis. So this is the first homegrown TB drug candidate which has shown efficacy in the TB patient. There has been not a single molecule that has come from India so far which has shown efficacy in the TB patients. So this is the first time in Indian history that we are seeing this. Thanks to our collaborators from TB Alliance and the Bill and Melinda Gates Research Foundation, this we hope will become the first drug from India for tuberculosis which will treat patients from India. So the take-home message I want to leave you with is that drug-resistant TB is a significant cause of treatment failure, and there is a definite need for new drugs to be discovered for tuberculosis. World Tuberculosis Day, which happens on 24th of March every year, we believe that we can end TB, and FNDR would be one of the organizations at the forefront of doing this. Thank you very much for your kind attention. Happy to take questions.
So thank you for the fantastic presentation. It's a proud moment to hear from very humble roots FNDR has started. They were bootstrapped, both of you. Bootstrap means with their own money they started it. They had a small grant from AstraZeneca, and it's just pure persistence. More than the TB persistence, it's these two scientists who have their perseverance and persistence that has got the first potential TB, anti-TB molecule from close to Puttaparthi, which is just 2 hours from here in Bangalore. And before we open it up for questions, sir, would our students get an opportunity to do internship or do projects with you, or get collaborations with you?
Absolutely, right? So this is something that we encourage significantly, is partnership and collaborations. So if any of you want to work in infectious diseases, not only tuberculosis, right? Whether it is gram-positive, gram-negative, or tuberculosis, or any BSL-3 level pathogen, right? We would be happy to look at you guys coming in and doing an internship. We are a very small unit, so there is limitation in terms of the number of people that we can accommodate, but definitely we can look at accommodating at least 1 or 2 students as part of an internship. What we also extend is that if a student is doing any research in any of these infectious diseases, all of the biology work, the in vitro work, can be done free of cost at our center, right? So,
to add to you, sir, we have a BSL-1 and BSL-2 facility here. We will show you shortly. We do have state-of-the-art facility. We have one of the largest AMR biobanks, nearly 10,000 clinically isolated escape pathogens, not the TB ones, because we don't have the BSL-3 facility here. So we would be happy. Many of our students do work on infectious diseases, especially in the field of antimicrobial resistance, and they would be happy to work on viral infections, fungal infections, and all the drug-related work. So
one other thing that we do is to organize site visits, right? So if any of your faculty or students want to come and see what an infectious disease research unit looks like in a pharma industry, we are a miniature one, I must confess. AstraZeneca was 75,000 square feet, we are 10,000 square feet. But regardless of that, we are fully self-sufficient and self-capable. So we've built a miniature AstraZeneca in FNDR, and I believe that anybody who wants to do research in infectious disease, which is a dying art today, especially drug discovery for infectious disease, we would encourage you to come and visit us.
Thank you very much, sir. Any questions now?
Thank you for a very wonderful, informative presentation. The first question I have is that the drug that you have developed, can we convert that into a transdermal patch? and then use it for pediatric patients, or like in the form of probably a syrup or something, and try it? Because one of the reasons why we are not able to give them drugs is because of the amount of tablets and the injections, all of it. So do you have that in your mind? That's the first one.
Absolutely. Let me answer that first, right? So doing a transdermal patch or any form of dosage form which could potentially increase the length of the activity of the molecule, right, without having to give repeated administrations, is very welcome. So that's why I put up a long-acting injectable. Transdermal patch is also possible. There are certain challenges with transdermal patches, but I think when you are giving drugs in combination, it's much easier to make them into oral or injectable format rather than a transdermal patch. But I'm not saying it's not possible. Definitely something worth exploring.
Thank you so much. The second one is, I know you have also worked with machine learning based drug discovery. So one of the problems that I see when it comes to pathogen-based diseases of how they are being mutated in different parts of the world, and then we get the mutation studies based using deep learning techniques, and then do a drug targeting or drug screening. Wouldn't that be possible for us to find a cure sooner than what we expect?
Again, a very, very, very valid point. I think the main challenge is access to data, right? Any AI model that you develop has to train on data, and data has to be of a certain quality, right? So when you look at publications, and Professor mentioned that not always data is reproducible, right? You have data being done in the West, data being done in the East, and they don't match. There are lab-to-lab variations that you encounter. So the data that goes into an AI/ML algorithm is definitely needs to be of a certain quality, right? And certain homogeneity when it comes to the different parameters that one looks at. So definitely is possible. So there is a database called, if I'm not mistaken, antibio database, which basically gives you a variety of MICs across the board for several antibiotics that have been developed across the globe. So this is global data. So you could use things like that to actually train your model and make them much more efficient. But again, tuberculosis is a very different beast. So you can't use a gram-negative data to train an algorithm for tuberculosis. You will need tuberculosis data to train the model, but definitely possible.
Thank you so much.
Thank you. Can you get up and speak?
Saram sir. So when you mentioned that if a patient has been taken drugs throughout the course, still there is a relapse rate of 6%, right, sir? So could it be that these tuberculosis are persistent tuberculosis?
That's probably the reason why, right? So there is persistent tuberculosis. There are certain persistent cells which escape the treatment and linger around, and that is what causes the relapse.
Thank you.
Hello, good afternoon, sir.
Yeah.
So in the introduction, I also heard that FNDR is also working on diabetes. So if it's yes, then what are the areas it is focusing on?
We don't work on any metabolic diseases at all.
Yes,
Our
sir.
focus is primarily on infectious diseases. The only place where we look at certain, comorbidities is other respiratory conditions. So we are interested in diseases like COPD, chronic obstructive pulmonary disease, and the interaction of that with tuberculosis. But we don't generally look at metabolic diseases and tuberculosis.
Yes, sir. Thank you, sir.
So coming to one question, if I'm
Sure.
permitted to ask. So long back, I read in a Nature paper or a Cell paper that tuberculosis goes and hides in stem cells which are quiescent, because they are non-dividing, they are not accessible to drugs, so the drugs do not work. So with regard to your probable candidate molecules, have you tested them in those kind of quiescent environments?
So we have not tested them specifically in cells which hide inside the stem cell, TB bacilli which hide inside stem cells, but what we have looked at is a variety of intracellular situations, and in all of those situations our molecule is able to work. So it's able to cross multiple barriers, enter the cell and work on the tuberculous bacteria. So definitely we believe that with a good pharmacokinetic properties and tissue distribution, if it reaches areas where the stem cells are present, definitely it will work there.
Okay. So next question.
Sairam, so first of all, very wonderful presentation.
Thank you.
And it is an honor to be listening to you. So I have a very simple and general question. Sir, what pros and cons should a student be having before choosing any particular field of study?
So that's a very tough question. I think there are multiple pros and cons, right? And it doesn't matter what field of study you choose. As long as you have a single-minded focus of excelling in that field, you will succeed. Whether it is tuberculosis research, whether it is gram-negative research, whether it is diabetes or whether it is obesity. Everything goes through a cycle, right? Today, obesity is the buzzword. Anywhere you go, people are talking about GLP-1 receptor agonists, yeah? Nothing else. I think it's a cycle. It will change. There are things like infectious diseases which are always a constant. It doesn't matter whether diabetes comes or goes, infectious diseases are there to stay, right? Whether it is viral infections, bacterial infections, or fungal infections. In fact, the importance of infectious diseases grows as some of these lifestyle diseases become more common, right? The incidence of fungal infections in diabetic patients, especially you saw that during COVID, mucormycosis gaining importance. The incidence of bacterial and fungal infections in cancer-treated patients, right? Significantly high. So I would say that as a student, it doesn't matter whether you choose to work in infectious diseases, oncology, metabolic diseases, whatever you do, I think as long as you focus on it and do good research, you will succeed.
Thank you so much, sir.
Sorry. I've got one more question. So you mentioned that you have been, the tests been done, trials been done using animal models. How about organoids? Like, wouldn't it be very much informative if we study from the development stage using the drug so that, we'll get a clear idea of how the drug is working from the development stage towards the end. And we could even potentially see the resistance, the patterns of resistance with different strains. Is that what is stopping us from doing?
So if you look at TB models in specific, right, and I contrast them to your gram-positive about 3 days animal models, right? 72 hours you have your result, maximum 5 days, right? The TB model goes on for at least 3 months. So you need to have a biosafety level 3 facility which can house the TB animals for 3 months. So there's a limitation in terms of the capacity with which you can do this work, right? You can't run multiple models over and over again. If you go to sterilizing animal model, and I didn't show data with the sterilizing animal models, which means that you're taking care of even the latent bacteria, and there is no relapse when you challenge with any immune suppressant agent, right? Those models go on for 9 months. So you're housing the animal in a biosafety level 3 facility for 9 months, which is a huge cost, right? So for a foundation like ours, it's a bit of a challenge to do these long-term animal models. But we are supported through government grants, variety of government grants from BIRAC, DBT, variety of sources, which helps us do a lot of this work. I would say that the animal models which are long-term is probably your best answer to look at drug resistance, to look at how the drug can be used in humans also as a treatment regimen, but those are extremely cost prohibitive.
Thank you. For want of time, because you're all waiting for lunch, we'll now take a break. So, see, when Swami says you have to serve the society, you have to do, these are the two people, he and Dr. Shandil, who left their lucrative job and decided that there is a lack of research in infectious diseases research, especially, and drugs for infectious diseases, and they have taken it head on, and it's been nearly 1.5 decades now, or 11 years.
11 years.
11 years. since 2014. It's not easy, especially being private, not for profit. Very challenging. There are global competitors everywhere with larger funding, and still they have put up. So basically we are seeing someone who is living Swami's message. Once again, please give a big hand.
I want to end with a couple of notes. One is, people like Swami are people who inspire others to take true service paths, right? And we are trying to do our small bit. I don't think we will ever reach what Swamiji has managed to do in terms of building education institutions, building hospitals, building all of the wonderful things that has been done. I don't think we are at that stage ever, but we are doing our small bit. And I think family plays a very important role. And today is the first time since I've started my career, and we've been married, that my wife is in the audience, right? When I told her I'm going to go give a lecture at the institute which Swamiji has established, she said that she wanted to come. She has never told me this. This is the first time that she expressed the interest, and today I'm really fortunate to have her. She's a microbiologist in her own right, and one of the molecules that we put up there, FNDR 20364, she probably isolated that when it came from a sample from Antarctica. She's a fermentation microbiologist. So thank you very much for your...
Since both Professor Roland as well as Dr. Sridhar, this is the first time you're here, just very briefly, all the institutions that are founded by Swami Bhagavan Shri Satya Sai Baba are 100% charitable. We do not charge a single penny from any student whatsoever for the education. There is absolutely no billing counter in our university. Even accommodation is provided free of cost in the hostel. They only have to pay for their food, which is like 5,000 rupees per month, which is nothing. If we go to a restaurant, we spend that much in a day when a group of us go there. So, and hospital is absolutely world-class treatment, totally free of cost. So what we have common is that we are passionate, we want to serve the society, we want to do things that have real impact on the society. So we would be happy to be associated with you long term, and we want to take these collaborations, take these interactions a lot more, in more ways and multiple times. Thank you. Let's pray that this is only a beginning for our continuous journey together. Sai Ram.
Thank you so much, Dr. Sridhar Narayan, for such a wonderful insight on the topic, and thank you, Dr. Pradeep, for combining science and spirituality and giving us such a wonderful outline of it at the end of the day. I now request Dr. A. Ashok, Department of Biosciences, and Dr. Vijay Kumar, sir, to please come up on stage and felicitate Dr. Sridhar Narayan. Thank you, sir. Sai Ram, everybody. We will now be breaking for lunch. I request the faculty of Sri Satya Sai Institute of Higher Learning ATP campus, along with the sisters of the campus, to please exit through the back door on my left, directly into the foyer for a lunch session. The boys can wait until the sisters have left. Sairam.